Good afternoon, everyone. I'm Felina. Welcome to our Industry Training Workshop Day 2 on the management of nitrosamine impurities in therapeutic products. Before we start our workshop proper, we would like to remind you that this presentation is intended to provide general guidance. Every effort has been made to ensure that the information is up to date and factual. We do not, however, warrant its completeness. So for specific concerns or questions, we recommend that you consult us directly. The aim of the training workshop today is to 1. Highlight key aspects of our recently published question and answer document that guides product registrants on managing nitrosamine impurities in therapeutic products. This document clarifies HSA's current approach, recommendations, and regulatory requirements pertaining to this issue. We encourage you to look at the document if you haven't had the opportunity to do so. And 2, we also aim to address some common concerns that you may encounter when managing nitrosamine impurities in your products. As you may remember, we conducted a survey from July to August 2024 to gather your inputs on the topics that you would like us to cover in this workshop. Respondents to the survey has expressed keen interest in several key areas. Foremost among these were risk mitigation strategies, closely followed by guidance on conducting risk assessments. There was also significant interest on the topic of establishing acceptable intakes, conducting confirmatory testing, understanding root causes and risk factors, as well as analytical method development. Therefore, this training workshop has been designed to address these topics. Our goal is to equip you with the knowledge and tools you need to manage nitrosamine impurities in your product. This is the agenda for today. Together with my colleagues, Sun Lan from the Therapeutics Products Branch and Dr. Zelwin from our HSA Pharmaceutical Laboratory, we will bring you through the introduction, risk factors and root causes, acceptable intake, risk mitigation strategies, current regulatory approach for new applications, current regulatory approach for registered products, and the analysis of nitrosamine impurities. I will start with the introduction. In this section, I will introduce the main goals of managing nitrosamine impurities in therapeutic products, bring you through a brief timeline of events, revisit the 3 steps approach in reviewing nitrosamine risk that we have introduced in 2020, show you the HSA nitrosamine resources that are available, and provide you with an overview of the international collaborative efforts in which HSA is currently participating. In managing nitrosamine impurities in therapeutic products, we aim to achieve a balance between safeguarding patients' health and to maintain supply of medicines to them. The need to safeguard patients' health stems from animal studies suggesting that nitrosamines are likely carcinogenic to humans. And to safeguard patients from unnecessary exposure, nitrosamines in medicines should be either eliminated or reduced to the lowest possible levels. However, on the other hand, we also know that this issue has proven to be complex and dynamic, affecting numerous products and involving more than over 100 nitrosamines and mostly novel to date. Addressing this issue requires substantial time, resources, and effort. Thus, as we work towards resolving this issue together, we must also ensure that patients continue to have access to the medicines that they need. Now, let me bring you through a brief timeline of events. In this slide, you will see some boxes in red and some in blue. Those in red are the regulatory measures that HSA have implemented to address the evolving events that have occurred over time that are depicted in the boxes in blue. This whole nitrosamine issue started in 2018 when nitrosamine was first detected in valsartan drug substance by a manufacturer. Over time, various nitrosamines in different types of angiotensin II receptor blockers, also known as ARB, across multiple manufacturers were discovered. These ended up in certain ARB products being recalled in 2019 due to the presence of nitrosamine above the acceptable level. As a result, HSA started to request for nitrosamine risk assessments for registered ARB products and in all new ARB-related applications. In late 2019, further recalls were conducted for other non-ART products like ranitidine and metformin due to the presence of nitrosamines above the acceptable level. This resulted in HSA to request all product registrants to conduct nitrosamine risk assessments in all their registered therapeutic products and in products that are identified as high risk for nitrosamines in new applications since 2020. To support the industry, HSA also published test methods for certain nitrosamines on our website to aid in method development and testing. 2022 saw the discovery of nitrosamine drug substance-related impurities, also known as NDSRIs. These are impurities that are structurally like the drug substance. They are mainly formed from reaction of amines present in the drug substance structure or drug substance fragments with trace levels of nitrites in excipients. As these nitrosamines are newly identified, there is scanty information on them. They do not have an established acceptable intake for us to work on, and thus confirmatory testing were in limbo. A lot of time had to be spent on deriving the acceptable intake for each of these NDSRIs. Hence, to address the need for establishing acceptable intakes for these novel nitrosamines at a faster rate, the carcinogenic potency categorization approach, also known as the CPCA, was developed by the global regulators. It is an approach for assigning a nitrosamine impurity to a predicted carcinogenic potency category with a corresponding acceptable intake. This is based on an assessment of the activating and deactivating features that are present in the structure of the nitrosamine. Activating structures are associated with an increase in carcinogenic potency and vice versa for deactivating structures. Given the emergence of NDSRIs, we also begin to see that an even larger number of products may potentially be at risk for nitrosamine, and thus HSA began requiring nitrosamine risk assessments for all chemical, new and generic drug applications at the point of screening. Together with other regulatory agencies, a scientific paper providing insights on risk factors, potential root causes, and recommendations for risk mitigation and control strategies was published. We also released a Q&A document on managing nitrosamines in therapeutic products that can be found on the HSA website. If you haven't had the opportunity to look at the Q&A document, its related appendixes, and the scientific paper, we strongly encourage you to do so. Overall, the nitrosamine issue has undergone significant evolution. In 2018-2019, it was initially perceived as an isolated problem affecting only a few products. However, as time progressed, it became clear that this issue impacts a wide range of products. And in response, HSA has implemented various regulatory controls to address this expanding concern. In 2020, when we requested all product registrants to conduct nitrosamine risk assessments for all their registered therapeutic products containing chemically synthesized drug substances, we introduced the 3-steps approach to review the nitrosamine risk. This 3-steps approach involved risk identification, detailed assessment, and risk mitigation. I will go into detail on this 3-steps approach in the next slide. We would like to highlight that this review is not a once-off exercise, but it is an ongoing process that should be occurring throughout the life cycle of the product. Let's now go into detail of the 3-steps approach in reviewing nitrosamine risk. In step 1, risk identification, we are required to conduct risk assessment to identify any risk for nitrosamine in drug substance or drug product manufacturing process. If no risk is identified, HSA does not need to be notified. However, please retain the risk assessment documents and submit them upon our request. If a risk is identified, we are required to notify HSA via online product defect reporting form or via email at hsa_productdefect@hsa.gov.sg together with the risk assessment documents. In step 2, detailed assessment, we are required to conduct detailed assessment to identify the root cause, carry out product testing, and develop appropriate CAPA where needed. We are also required to submit the detailed risk assessment report that should include the investigation outcome, test results, and CAPA to HSA. We will then discuss the appropriateness of the proposed CAPA. In step 3, Risk mitigation. We are required to submit the appropriate variation application for the required changes to mitigate the nitrosamine risk. Now that we understand that this whole process is not a once-off exercise but an ongoing process throughout the life cycle of the product, and given the dynamic nature of this whole issue, it is crucial to continuously monitor developments and conduct reassessments with discovery of any new root causes or implementation of any changes that could potentially alter the nitrosamine risk of the product. This brings us to a question that we received in yesterday's training workshop. The participant asked whether detection of an NDSRI above the acceptable limit is considered a product defect, and if a recall is warranted. The answer to the first part of the question is yes. The detection of a nitrosamine in the product is considered a product defect, and HSA needs to be informed. More so in this case, where the NDSRI was detected above the acceptable limit. As for the second part of the question on whether the product needs to be recalled, we'll go through these at the later part of the workshop. We would like to draw your attention to our HSA nitrosamine webpage accessible via the HSA website. This slide offers an overview of the various sections that are available on the webpage. The main page offers general information on the background and outlines a regulatory approach on this nitrosamine impurities issue. It also includes links to 3 subpages to provide more information on the list of impacted medicine as well as the guidance and test methods that we have developed. Let's move on to take a look at the 3 subpages. In the first subpage, List of Impacted Medicines, you will see 2 tables. Table 1 provides information on the list of therapeutic products recalled due to nitrosamine impurities, while Table 2 presents the list of drug substance for which HSA has temporarily allowed the use of a higher interim acceptable intake to avoid supply disruptions to patients. These lists are dynamic and will be updated to incorporate the latest information as and when there is any. Second subpage, Guidance for Product Registrants, outlines required actions for product registrants in managing this issue. It is also where you can find our Q&A document and its related 5 appendixes. The Q&A document provides further details and clarifies on HSA's current approach, recommendations, and regulatory requirements for managing nitrosamine impurities in therapeutic products. Appendix 1 shows HSA recommended acceptable intake for certain known nitrosamines. Appendix 2 is an assessment aid for assessing nitrosamine risk. Appendix 3 is a guidance on submission of nitrosamine risk assessment where risk is identified. Appendix 4 outlines the carcinogenic potency categorization approach (CPCA) for nitrosamines. And Appendix 5 outlines the enhanced Ames test conditions for nitrosamines. We will be going through Appendixes 2 and 3 in more detail later in the workshop. Appendixes 4 and 5 are straightforward and self-explanatory. We encourage you to look at the documents if you haven't had the opportunity. The third subpage presents test methods that HSA has developed for detecting and quantifying nitrosamine impurities in certain products that you can adapt for the testing of nitrosamines in your own products. We would like to emphasize that these methods must be validated by you before use to ensure that they suit your specific needs. While not exhaustive of all nitrosamines, we will update this webpage to share new analytical methods for nitrosamines as we develop them. In terms of international collaboration, HSA collaborates with regulatory bodies from Australia TGA, Brazil ANVISA, Health Canada, ADQM, EMA, Japan MHLW, Swissmedic, the US FDA, and the WHO as part of the NISG or NITWG. This international collaboration strives to harmonize the regulatory approaches for risk assessment, scientific principles for determining acceptable intakes, and risk mitigation strategies wherever possible. While we strive for alignment, it is also important to note that occasional divergence in actions taken may be unavoidable due to various factors, including the availability of similar products in the different markets. This collaboration has led to the development of the scientific paper, Regulatory Experiences with Root Causes and Risk Factors for Nitrosamine Impurities in Pharmaceuticals. This paper provides an insight into the current scientific understanding of risk factors and potential root causes for nitrosamine impurities from a quality perspective, as well as recommendations for risk mitigation and control strategies. We highly encourage you to look at the documents if you haven't had the opportunity to do so. Now, I will pass to Stelan, who will bring you through the next section of this workshop.
In this presentation, I will outline the common risk factors and root causes associated with nitrosamine formation in both drug substance and drug product processes. Secondly, I will discuss the acceptable intakes accepted by HSA and a few approaches to establish the acceptable intakes. Thirdly, we will explore risk mitigation strategies, focusing on practical measures which manufacturers can implement to reduce nitrosamines in their processes. And finally, we will go through the regulatory approach for new product applications, highlight information that we will expect to see in a comprehensive risk assessment. Our aim is to provide clarity on our regulatory requirements and guide product registrants to relevant resources on nitrosamine management. As a recap, nitrosamines are a class of compounds defined by their N-N-double-bond-O functional group. In the ICH M7 guidelines, the nitrosyl group is identified as a chord of concern. Some nitrosamines are classified as Group 1, 2A, or 2B by the International Agency for Research on Cancer, IARC, underscoring their potential health implications. Nitrosamines are impurities, and similar to other impurities addressed under ICH Q3, there should be adequate control strategies in place. The regulatory framework for managing nitrosamine impurities is designed to complement the existing guidelines on impurities. Nitrosamines are formed by a nitrosating agent in the presence of a vulnerable amine. Understanding this reaction mechanism can help us identify the risk factors in the process. In 2018, the presence of N-nitrosyl dimethylamine, NDMA, was found in valsartan-containing products, and the root cause was linked to specific manufacturing conditions. It involved the use of dimethylformamide DMF as a solvent and the presence of sodium nitrite, which is commonly used as a quenching agent to neutralize azide. Trace amount of dimethylamine present in the DMF solvent underwent nitrosation when exposed to the sodium nitrite, resulting in NDMA formation. Nitrite sources are not limited to sodium nitrite. They can be found in other reagents such as nitric acid or present as contaminants in azides. Similarly, amine sources are diverse and can be present in reagents, solvents, starting materials, or intermediates. Nitrosamine formation is not solely limited to external factors in the manufacturing process. In some cases, it can be attributed to intrinsic properties of the drug substance itself. A prime example is ranitidine, which contains both an amine group and a nitrosyl source within its molecular structure. This unique combination makes ranitidine and other compounds with similar structures susceptible to fragmentation under specific conditions to produce NDMA. There has been recent emergence of nitrosamines formed in drug products, termed as nitrosamine drug substance-related impurities, or NDSRI for short. These are formed from vulnerable amine structures in drug substance or drug substance fragment reacting with trace levels of nitrites present in the excipients. There are few scenarios where vulnerable amines can be present. Firstly, a secondary amine structure can be present in the drug substance structure itself. We see this in beta blockers such as propranolol, bisoprolol, as well as ACE inhibitors like quinapril and lisinopril. Drug substances with tertiary amines can undergo dealkylative pathways to produce secondary amines, for instance, sumatriptan. Some drug substances do not contain secondary or tertiary amines, but they can break down to form degradation impurities that do contain secondary amines. An example is sitagliptin. Nitrites are present in a variety of excipients, and they serve as a primary source of nitrosating agents to react with amines in the drug substance or drug substance fragments, even if present in trace amounts. Different excipients contain varying levels of nitrites as expected. However, the same excipient from different suppliers may contain nitrite at different levels depending on the manufacturing process. Nitrite levels can also vary from batch to batch even when purchased from the same supplier, adding a layer of complexity to risk evaluations. When formulating a control strategy for nitrosamine, manufacturers should consider the established acceptable intakes. Appendix 1 contains a list of acceptable intake levels for certain known nitrosamines as recommended by HSA. It is important to note that this list is not exhaustive, and the inclusion of a nitrosamine on this list does not necessarily indicate the presence of nitrosamine impurities in all products or formulations containing the drug substance. So please refer to the current version of the document as it will be updated as more information emerges. Appendix 1 shows the acceptable intake in nanograms per day. The acceptable limit in ppm can be calculated by dividing the acceptable intake in nanograms per day by the maximum daily dose of the drug substance in milligrams. For example, NDMA has a recommended acceptable intake of 96 nanograms per day. The drug substance metformin has a maximum daily dose of 3000 milligrams per day based on a certain package insert. Therefore, the acceptable limit is calculated by taking 96 nanograms per day divided by 3000 milligrams per day to obtain 0.032 ppm. If the acceptable intake of the nitrosamine is listed in Appendix 1, then we will follow Appendix 1. If it is not listed in Appendix 1, then we will consider whether it is published by other reference agencies. If yes, the acceptable intake published by reference agencies can be proposed. If no, we will consider whether the nitrosamine has substance-specific animal carcinogenicity data. If yes, the TD50 can be calculated to derive a limit as per ICH M7. Otherwise, the following approaches can be taken. If the nitrosamine does not have substance-specific animal carcinogenicity data, the acceptable intake can be established using one of these four approaches: CPCA, the Enhanced Ames test, in vivo mutagenicity study, or using a surrogate nitrosamine with robust carcinogenicity data. The CPCA approach is described in Appendix 4, and the Enhanced Ames test is described in Appendix 5. If the Enhanced Ames test is conducted and is negative, then an AI of 1.5 micrograms per day is applicable. If the Enhanced Ames test is positive, the AI should be established using the CPCA or surrogate nitrosamine approach. If an in vivo mutagenicity study is conducted and is negative, the nitrosamine can be controlled as a non-mutagenic impurity as per ICH Q3A or B. If the in vivo mutagenicity study is positive, the AI should be established using the CPCA or surrogate nitrosamine approach. Earlier we have discussed how nitrites in excipients can react with amines from the drug substance or its fragments to form nitrosamines in drug products. However, we have noticed that this risk is often overlooked in the assessments that we review. Some risk assessments lack crucial information, particularly regarding the risk of nitrosamine formation in the drug product itself. To address this gap, we have developed Appendix 2, an assessment aid designed to help product registrants more accurately identify nitrosamine formation risk in both the drug substance synthesis and drug product manufacturing processes. While using Appendix 2 is not mandatory, we strongly recommend it as a tool to ensure your risk assessment is comprehensive. The key is to conduct a thorough evaluation whether you use our aid or your own method. The drug substance section of the questionnaire is designed to help identify specific risk factors in the manufacturing process. You'll be prompted to consider potential sources of nitrites and amines, which could be present in the form of reagents, solvents, raw materials, etc. While sitagliptin itself does not contain a vulnerable amine in its molecular structure, it can degrade to form a degradant with a secondary amine. To reflect this, we will mark yes to question number 3 about the presence of an amine. For the sake of simplicity in this example, the rest of the questions are marked as no. However, in the actual assessments, you should carefully consider each of the questions. If your DS process contains any other risk factors, make sure to mark those as yes accordingly. This section is designed to identify NDSRIs associated with the drug substance. It also prompts the user to consider residual nitrites in the excipients. In our sitagliptin example, we have marked yes for points 1, 2, and 3 to reflect this risk. Reviewing the overall risk, the amine degradant identified earlier could react with residual nitrites in the excipients to form a nitrosamine. Indeed, according to Appendix 1, a specific nitrosamine called the NTTP has been identified in sitagliptin products. Some NDSRIs have been observed to increase during long-term storage or stress testing. In this example for sitagliptin, the precursor is formed via a degradation pathway, hence it should be assessed whether the corresponding nitrosamine will increase during storage. Identifying the risk factors provide a foundation for implementing effective risk mitigation and control strategies. Here we introduce some common strategies that manufacturers can consider when designing the drug substance process. When choosing raw materials, avoid the use of amines or amides where possible. Residual nitrites in reagents used should be understood and controlled where appropriate. During process design, ensure the upstream nitrites are effectively purged before introducing downstream amines. If the formation of nitrosamines is unavoidable, ensure effective purging process is in place. To control cross-contamination within the facility and between process steps, effective cleaning procedures should be in place to remove any residual nitrites from the previous process. The use of recovered materials should be limited as much as possible. Drug product manufacturers can consider controlling nitrite levels in the excipients in these few ways. During formulation design, select excipients with lower nitrite content. Conducting a well-designed drug excipient compatibility study can guide the selection of suitable excipients. Choose excipient suppliers with robust nitrite control measures and implement comprehensive supplier qualification program. Where required, include a test parameter for nitrite in the excipient specification. The process and formulation design can be optimized in a few ways. Some unit operations, such as wet granulation and fluid bed drying, are known to facilitate the nitrisation reaction. The process parameters for these unit operations can be optimized to reduce impurities formation. Consider incorporating nitrite scavengers, such as ascorbic acid, propyl gallate, in the drug product formulation, as studies have shown their potential to inhibit nitrosamine formation. Manufacturers are expected to implement appropriate control strategies to ensure nitrosamine levels remain within the acceptable intake throughout the product's shelf life. A test for nitrosamine should be included into the product specification where applicable. Routine testing is required if the risk of nitrosamine formation is high, or if levels exceed 30% of the acceptable intake, or if the root cause is unknown. If routine testing is performed, then the limit of quantitation, LOQ of the test method, should minimally correspond to the acceptable intake. Skip testing can be proposed if the root cause is known and nitrosamine levels are consistently at or below 30% of the acceptable intake. To justify for skip testing, the LOQ should minimally correspond to 30% of the acceptable intake. Testing for the nitrosamine can be omitted from the specification only if the root cause is known and levels are consistently less than or equals to 10% of the acceptable intake. Appropriate measures should be in place to ensure that the nitrosamine level will remain below 10% throughout the product shelf life. To justify for total omission of the specification, the LOQ should minimally correspond to 10% of the acceptable intake. Appendix 2 can be used as a guide to identify and assess any potential nitrosamine risks. If no risk is identified, submit only Appendix 2. If a risk is identified, then a detailed risk assessment should be performed using Appendix 3 as a guide. The detailed risk assessment should include information such as the identity of the nitrosamine, possible root causes, acceptable intake for the nitrosamine identified, test method validation report, test results, as well as risk mitigation and control strategies. The same risk assessment that is submitted to other regulatory agencies can be used, provided that the information is relevant to the locally registered products. Other points to note regarding risk assessments for new GDA, NDA, and relevant MIV applications. The nitrosamine risk assessment is required for all chemically synthesized drug substance. It must be provided at the point of submission for any new GDA and NDA applications, and any MIV where the change made could alter the nitrosamine formation risk. It is submitted as part of the CTD S3.2 drug substance section or CTD P5.5 in the drug product section. The risk assessment should include confirmatory test data when nitrosamine risk is identified. For products with high risk for nitrosamine formation, test results are required before the application can be accepted for evaluation. The level of nitrosamines present should meet the AI specified in Appendix 1 with the necessary controls in place where required. I will now pass it back to Felina, who will bring you through the next section of this workshop.
I will now bring you through this section on current regulatory approach for registered products. We will cover the risk assessment requirements for registered products, look at the similarities and differences between the requirements for new applications and registered products, as well as touch on the interim acceptable intake. Let us start off with the risk assessment requirements for registered products. Firstly, risk assessment is required for all therapeutic products containing chemically synthesized drug substances. Secondly, you can use Appendix 2 and 3 to help identify the risk for nitrosamine formation and to guide your risk assessment. The same risk assessment that is submitted to other regulatory agencies can be used, provided that the information is relevant to the locally registered products. So for example, they must have the same registered manufacturers, product formula, storage conditions, and specifications. Do these requirements sound familiar? I hope they do, because these are the same requirements that Celine has presented earlier for the new applications. The nitrosamine risk assessment for registered products actually do not differ significantly from that for the new applications. They are largely the same. Let me highlight the key differences in the risk assessment requirements for registered products and new applications. For new applications, risk assessment should be provided at the point of application submission regardless of the risk present. For registered products, HSA needs to be informed if a risk is identified during the initial and reassessments. HSA can be notified via online product defect reporting form or via email at hsa_productdefect@hsa.gov.sg with the risk assessment documents containing the root cause, confirmatory test results, interim or final CAPA plan, health hazard assessment, an assessment on whether the product is medically necessary or important, market action proposed if any, an assessment on whether any disruption to product supply is expected. If no risk is identified, HSA do not need to be informed. However, please retain the risk assessment documents and submit them upon request. Another key difference would be that for registered products, we will need to consider if market actions is required if the test results exceed the acceptable intake, as they may already be in the market, and we do this by balancing the risk to patient safety, the medical need of the product, and the market availability of the product. As with all other product defects involving impurities, investigate the root cause, establish and implement CAPA to reduce the nitrosamine levels to less than or equals to the acceptable intake. This is to prevent unnecessary long-term exposure and safety risks to patients. This now brings us back to the question yesterday on whether a product that's found to contain a nitrosamine above the acceptable limit needs to be recalled. Please note that not all product defect will end up with a recall. An assessment needs to be made considering the above to determine whether recall is warranted. This brings us to the next key difference between our approach for registered products and new applications. We acknowledge that CAPA investigation and implementation needs substantial time. Hence, to prevent disruption of supply of registered products, particularly essential medicines, an interim acceptable intake can be proposed for consideration. Please contact us to discuss further on whether the use of an interim acceptable intake is appropriate and whether the proposed interim acceptable intake is acceptable. Should you wish to propose an interim acceptable intake, please ensure that the approach you have chosen to derive it is scientifically robust and demonstrates that the theoretical excess cancer risk does not exceed 1 in 100,000 during the CAPA implementation period. Conversely, please note that an interim acceptable intake is generally not allowed for products in new applications. The level of nitrosamines present in products in new applications should meet the acceptable intake specified in Appendix 1 before the application can be approved. In this slide, we are going to focus on the most common way for deriving the interim acceptable intake, which is to use the less than lifetime approach or the LTR approach as described in the ICHM 7 guideline. For this method, the interim acceptable intake is calculated using the two most conservative adjustment factors, 6.7 and 13.3, depending on the medicine's treatment duration, as shown in the table on the slide. It is important to note that the acceptable intake should not exceed 1.5 micrograms per day, except in the following cases. The acceptable intake in Appendix 1 is already above 1.5 micrograms per day, or the nitrosamines classified as Category 5 according to CPCA, or the nitrosamine is shown to be negative in an enhanced Ames test. The application of these adjustment factors will not be expected to exceed the theoretical excess cancer risk of 1 in 100,000 during the period of CAPA implementation. You should complete the CAPA as soon as practically possible. Start promptly to buffer against potential unforeseen delays in your CAPA implementation process. Please provide us with the projected timeframe for completing the CAPA and we can discuss on the appropriateness of the proposed timeline. Here are some factors to keep in mind when using the interim acceptable intake. Time-limited use. The interim acceptable intake can only be used for a predefined period as agreed with HSA because it is only a temporary measure to ensure continuity of medicine supply while the CAPA is being implemented. Additional monitoring. Additional monitoring measures may be required during period of use to ensure nitrosamine levels remain within acceptable levels. For example, mandatory routine testing of every batch of drug product prior to supply in Singapore may be required. Applies to single nitrosamine only. The interim acceptable intake cannot be used for products with multiple nitrosamines. Please contact us to discuss on the approach to be taken for such cases. To conclude, nitrosamines are a subset of impurities that are routinely identified in CTD as 3.2 and P5.5. Therefore, like other impurities, they need to be evaluated during product applications and reported if detected in the product post-registration. In line with our standard product defect reporting procedure, please provide as much information as possible. Do not postpone your report if certain details are not immediately available. You can always submit additional information later when it becomes available. For confirmatory testing, please remember to test for products that are stored under our locally registered conditions and include batches that are near the end of their locally registered shelf life to provide some insights into the nitrosamine levels in products stored under our climatic conditions. Please note that information shared is current as of today. Given the dynamic nature of this issue, please keep abreast of the developments and conduct reassessments if new information arises that could potentially alter the nitrosamine risk of a product. We will move on to address some of the common questions that we have received from you all prior to this workshop. The first question is, Does the risk assessment exercise apply to biological therapeutic products? And the answer is, a nitrosamine risk assessment is still required to be submitted to HSA if nitrosamine risk factors are present. So for example, it should be conducted for biological therapeutic products containing chemically synthesized fragments such as antibody drug conjugates. The next question, can HSA align the acceptable intake and risk assessment template with other regulatory agencies? Continual harmonization efforts through international collaborative activities are ongoing. HSA collaborates closely with regulatory bodies from Australia TGA, Brazil ANVISA, Health Canada, EDQM, EMA, Japan MHLW, Swissmedic, the US FDA, and the WHO as part of the NISG and NITWG to harmonize as much as possible the regulatory approaches for risk assessment and the scientific principles for determining acceptable intake. The regulatory approach in terms of risk assessment requirements as well as establishing of acceptable intakes for nitrosamines as listed in Appendix 1 are largely internationally aligned. For nitrosamines which are not listed in Appendix 1, product registrants may propose the acceptable intakes published by our reference agencies for discussion. Risk assessment templates that submitted to other regulatory agencies can be used, provided that the information is relevant to the locally registered product. Question 3. Can HSA provide flexibility and consider acceptance of product registrants' proposed acceptable intakes based on scientific or technical justification and data? Product registrants may propose alternative acceptable intakes, provided they are supported by robust scientific and technical justifications with relevant data. The acceptability of alternative proposals will be assessed and determined during the evaluation. So for example, the acceptable intake of N-nitrosodiclofenac was revised from 1.5 micrograms per day based on CPCA to 78 micrograms per day via structure-activity relationship using N-nitrosodiphenylamine as a reference. The revision was supported by showing that N-nitrosodiphenylamine can be considered as a conservative surrogate compound. Question 4. Given the challenges of finding a laboratory in Singapore to perform nitrosamine testing and the high costs associated with developing an in-house test method, how can product registrants overcome these challenges? HSA has been publishing the test methods that we have developed when available. Product owners or manufacturers may also refer to the available methods published by our reference regulatory agencies. These methods can be used as a starting point for the development and validation of the product owner's or manufacturer's own methods. It's important to validate the method before use to ensure its suitability for the intended purpose. While regulators can help by providing guidance for method development, it is still the responsibility of the product owner or manufacturer to ensure that adequate resources are available so that their products continue to meet safety, quality, and efficacy standards. Product owners and manufacturers are strongly encouraged to utilize these available resources to develop their own testing capabilities. Question 5. Given that confirmatory testing is time-consuming, it may impact marketing plans. Can product registrants initiate the submission process first so that HSA can start evaluation while awaiting the results of confirmatory tests? HSA requires the complete risk assessment and the test results where applicable at the time of application submission for the evaluation to be carried out in a timely and efficient manner. This is to avoid the scenario where the evaluation has been completed but the application has to be rejected eventually if the nitrosamine test results is above the acceptable intake, resulting in abortive work for both HSA and companies. However, where the product registrant is unable to meet the above requirements and the product is critical for meeting Singapore's healthcare needs, please contact HSA to discuss further. Question 6. Will the registration timeline be affected when a new nitrosamine is identified during evaluation? Can HSA approve the application first with the product registrant committing to provide test results post-approval? Product registrants are required to demonstrate effective, adequate control of impurities before product applications can be considered for approval. This approach applies to all impurities including newly identified nitrosamines. Hence, if the product has been assessed to have a high risk of the new nitrosamine, the final registration timeline for the application will be dependent on the time taken by the product registrant to provide the test results. This regulatory approach for new applications is necessary to protect public health by limiting the introduction of medicines with nitrosamine levels above acceptable limits into the market and to reduce the need for post-market interventions like recall. However, considerations may be given to products that have high medical needs in Singapore, where the need of the product is assessed to outweigh the potential risk for the nitrosamine. Question 7. Can product registrants omit data on nitrosamine formation under Zone 4B conditions if the information is unavailable due to the manufacturer primarily serving markets in a different climatic zone? For registered products, HSA would need to understand the risk of nitrosamines in these products as soon as possible, as the products are already on the market. Hence, HSA may accept other forms of justification if they are supported by robust scientific data. For example, by extrapolation from stress to accelerated testing. This will be subject to further assessment. For new applications, stability data at Zone 4B is required in accordance with current product registration guidelines. Question 8. Can HSA provide a longer timeline to respond to post-market queries? Extension to timelines can be requested provided that the reasons for the delay are well justified and there are no urgent matters requiring immediate action. HSA encourages product registrants to work together with their manufacturers to conduct the assessments promptly. Should more time be required, please submit the written request for an extension, clearly explaining the reasons for it. This is the last question. Why is the less than lifetime LDL approach for acceptable intake not applicable to new applications but only to registered products? As nitrosamines are generally highly potent and classified as a core concern in ICH M7 guideline, it was assessed that the LDL approach is not applicable to establish the acceptable intake. This approach is only considered as an interim measure to minimize supply disruption during CAPA implementation for certain marketed products. It is applied on a case-by-case basis to balance risk mitigation while ensuring continuity of medicines to patients. Now, I will hand it over to Dr. Zheng Yun from our HSA Pharmaceutical Laboratory, who will be sharing on the topic Analysis of Nitrosamine Impurities in Therapeutic Products.
Good afternoon. I'm Zheng Yun from PharmLab HSA. Today, I will be sharing the analysis of nitrosamine impurities in therapeutic products. My presentation covers the introduction of the analysis of nitrosamine impurities, the key consideration of the method development and validation. I will also share 2 examples during this part. The challenges and the future of nitrosamine impurities testing will be mentioned as well. Nitrosamines are harmful substances found as impurities in various therapeutic products. There are 2 main types of nitrosamine impurities: small molecular nitrosamines and nitrosamine drugs substance-related impurities, also called as NDSRA. Small molecular nitrosamines such as NDMA, NDEA, NMBA, NDIPA, NEIPA, and NDBA. They have simple structure and can be formed through chemical reactions. NDSRA are more complex and often formed during the manufacturing process of specific drugs. They have similar structure to the API and are generally unique to each API. Both types pose potential health risks, including carcinogenics. Factors like raw materials, manufacturing, and storage conditions can influence nitrosamine formation. Nitrosamine impurities analysis is crucial for safeguarding patients' health. Accurate analytical methods enable early detection of these harmful impurities in drug products. By identifying nitrosamine impurities, manufacturer can promptly take corrective actions, such as adjust manufacturing process or recalling affected products. As part of the regulatory approach to manage the risk of nitrosamine impurities in therapeutic products, the regulation agents, such as FDA, EMA, and HSA, have required all product registrants of therapeutic products containing chemically synthesized drug substances to conduct risk assessment of the product to identify any potential risk of nitrosamine impurities. Where potential risk is identified, confirmatory testing should be done, and the necessary risk mitigation measure must be implemented. Test methods for determining the presence of several types of nitrosamine in certain drug substances have been published by various regulatory agencies, including FDA, EMA, and HSA. This method can be used as the starting point for the development and validation of analytical methods for nitrosamine in other drug substances. The development of a reliable method for nitrosamine analysis requires careful consideration of several key factors. Sensitivity and specificity are essential to ensure accurate detection and quantification of nitrosamines at low levels. A low limit of detection is crucial for detecting trace amount of these compounds. Proper sample preparation, including sample cleanup, is necessary to extract nitrosamine from the sample matrix without interference. Analytical techniques such as LC-MS and GC-MS are commonly used for nitrosamine analysis due to their high sensitivity and selectivity. Method validation is essential to ensure the accuracy, precision, and robustness of the developed method. Factors such as linearity, accuracy, precision, robustness, and matrix effect must be evaluated during validation. By carefully considering these factors, a reliable and accurate method for nitrosamine analysis can be developed. Nitrosamines are often found in trace amount, requiring high sensitivity for accurate detection and quantification. Low LOD and LOQ values are essential. LC-MS and GC-MS offer excellent sensitivity due to their ability to detect and quantify substances at low concentrations. Instrument settings, such as ionization source and mass spectrometer settings, can be optimized to improve sensitivity. Proper sample preparation, such as cleanup, can also enhance the sensitivity. The method should be capable of simultaneously analyzing multiple analytes and eliminate sample interference. Sample extraction should be chosen based on the sample type, such as liquid, solid, and the formulation. Liquid-liquid extraction, solid-phase extraction, or direct dilution are common methods. Cleanup steps such as precipitation, filtration, or additional SPE might be necessary to remove interfering component. LC-MS and GC-MS are powerful tools for nitrosamine analysis, offering excellent sensitivity. LC-MS is suitable for polar compounds, reducing thermal degradation, but it can suffer from lower sensitivity and matrix effects. GC-MS, on the other hand, offers high sensitivity, specificity, and wide application, with relatively simple sample preparation. However, it is limited to nonpolar compounds and can cause thermal degradation. The choice between LC-MS and GCMS depends on the specific characteristic of the nitrosamine and the sample matrix. The choice of chromatographic color and morphophase is crucial for effective nitrosamine analysis by LC-MS. A reverse-phase column is often suitable due to its ability to retain polar compounds like nitrosamines. The morphophase should be carefully selected to ensure adequate separation between nitrosamine impurities and other sample components. A combination of aqueous buffer and organic solvent is commonly used. For complex samples, gradient elution can be employed to improve peak shape and resolutions. By optimizing the column and morphophase, analysts can achieve better separation and detection of nitrosamine impurities. For the MS conditions, electrospray ionization (ESA) is often preferred for LC-MS due to its sensitivity and compatibility with polar compounds. Atmospheric pressure chemical ionization (APCI) can be used for small molecular nitrosamines. Electron ionization (EM) is widely used in GCMS. Optimize source parameters like capillary voltage, cone voltage, and temperature for efficient ionization and fragmentation. Select appropriate multiple reaction monitoring transitions for each nitrosamine impurity based on their fragmentation patterns. Use internal standards to account for matrix effect and variation in recoveries. Matrix effects can arise from co-extracted components, interfering with NDSRI ionization or detection. Strategies to mitigate matrix effect include sample cleanup technique such as SPE, isotope dilution MEMS, matrix matched calibration standards, and using instrumental valve switching set to divert the main component to waste to eliminate the matrix effect. For example, during the analysis of NDMA in ranitidine products, the NDMA peak is very tiny compared to the huge ranitidine peak. We were unable to see any NDMA signal due to the severe matrix effect. In this case, we divert the majority of ranitidine to waste and able to quantify NDMA in ranitidine products accurately. This approach helps ensuring accurate quantification of nitrosamine in the presence of complex drug matrix. Chromatographic separation is crucial for accurate nitrosamine impurity analysis. With sufficient separation, we can divert the interfering components to waste. For example, during the analysis of nitrosol salbutamol in salbutamol products, the retention time difference between nitrosol salbutamol and the two main ingredients, salbutamol and ipratropium, are sufficient to allow us to divert the salbutamol and ipratropium to waste and mitigate the matrix effect. The key factors influencing the separation include column selection, morphophase, and proper setting of flow rate and temperature. By optimizing these parameters, analysts can achieve efficient separation and accurate quantification of nitrosamine impurities. A typical LC-MS method development workflow involves several key steps. First, a MS method is created for the target impurities. Next, a LC method is developed and evaluated to ensure effective separation of the nitrosamine impurities and API and other ingredients. The LC method should be optimized if the separation is not satisfied. Once the LC method is finalized, Bulk switching is incurred. A suitable sample extraction method is then developed, followed by sample analysis using the optimized LC-MS-MS method. This stepwise approach helps ensuring the accuracy and reliability of the developed method for nitrosamine analysis. Once the method is ready, a validation study was conducted according to SHQ2 guidelines to ensure the reliability of the analytical procedure. Key validation parameters include specificity, linearity, LOD, LOQ, accuracy, precision, and also the measurement uncertainty. It is essential to perform proper method validation or verification before adopting an existing method to ensure its suitability for the inadequate application. Here is an example of NDSRA analysis, the determination of nitrosulfuroxidine in fluoxetine products. Fluoxetine is a medicine used to treat conditions like depression, anxiety, and obsessive-compulsive disorder. It works by increasing levels of a chemical in the brain called serotonin. Nitrosulfuroxidine is a nitrosamine impurity found in fluoxetine products. It is a potent carcinogen compound. The allowed intake limit of nitrosulfuroxidine is 100 nanogram per day, while the max daily dose of fluoxetine is 80 mg. Therefore, the allowed concentration of nitrosulfuroxidine in fluoxetine product is 100 nanogram per 80 mg, which is equivalent to 1.25 microgram per gram. The limit of quantification should be much lower than this value. To address this challenge, an LC-MS-MS method was developed. The instrument used for quantification was Q-Trap 7500 LC-MS-MS. If nitrosulfuroxidine was detected in the product, a Q-Executive high-resolution MS instrument would be used for the confirmation. The method was validated to ensure its accuracy, precision, and robustness. This validated method can be effectively used to monitor the nitrosulfuroxidine levels in fluoxetine products. Here is the chromatogram of nitrosulfuroxidine and its internal standards, nitrosulfuroxidine D5. The retention time difference between nitrosulfuroxidine and fluoxetine was sufficient to divert the API to waste and effectively minimize the matrix effect during the analysis. The sample was prepared by extracting a certain amount of sample, equivalent to 50 mg fluoxetine, using 5 ml of mixture of methanol and water containing 10 nanogram per ml of internal standard. It was then vortex, sonicate, centrifuge, and filter. The filtration was used for the LC-MS-MS analysis. The results of the method validation were summarized in this table. The instrument LOD and LOQ are 0.4 and 1 nanogram per ml. The corresponding method LOD and LOQ are 4 and 10 microgram per gram. Another example is the determination of NDMA in metformin products by high-resolution accurate GC-MS. Both metformin and NDMA are small molecular compounds. The determination of NDMA in metformin products is challenged due to both compounds have similar solubilities. The allowed intake limit of NDMA is 96 nanogram per day, while the max daily dose of metformin is 3 gram. Consequently, the permissible concentration of NDMA in metformin products is 96 nanogram per 3 gram, which is equivalent to 32 nanogram per gram. The method LOQ should be less than this value. Hence, a highly sensitive analytical method is required. In our method, the high-resolution accurate GCMS instrument was used. It offers very good sensitivity and specificity. Method validation was done including the LOD-LOQ, linearity, accuracy, precision, robustness, and measurement uncertainty. The extraction used DCM and 1N-hydrochloric acid solution. Most of the polar compound of metformin was extracted into the aqueous layer and reducing the interference during the NDMA analysis. The results demonstrated that both immediate release and extended release formulation formed a well-separated layer following mixing and centrifuge, enabled the direct withdrawal of DCM layer for analysis without the need for additional purification steps. An internal standard, NDMA-D6, was added to mitigate the impact of the sample loss during the extraction and analysis process by adding a known amount of internal standards to the sample before extraction. Any loss incurred during the analytical workflow can be quantified and corrected, ensuring accurate quantification of the target analyte. Here is the general workflow for the determination of NDMA in metformin product by HRA GCMS. The method validation was done for these tests. The results were summarized in the table. The instrument LOD and LOQ are 0.5. The method validation was done for these tests. The results were summarized in the table. The instrument LOD and LOQ are 0.5 and 1 nanogram per ml. The corresponding method LOD and LOQ are 10 and 20 nanogram per gram. Regulatory agencies worldwide have established guidelines for nitrosamine impurity analysis in pharmaceutical products. These guidelines recommended analytical methods like LC-MS-MS and GCMS, risk assessment and quality control measures. The FDA, EMA, Health Canada, Japan Ministry of Health, and HSA have issued specific guidance emphasizing the importance of robust analytical method and risk-based approach to ensure the safety of medications. Nitrosamine analysis is often complicated. Matrix effects, such as interference and co-elution, can affect the detection and quantification of nitrosamine in samples. Data interpretation can be complex due to the presence of multiple peaks and potential interference from other compounds in the sample matrix. Additionally, limited availability of reference standards for the potential nitrosamine can pose challenges. Obtain nitrosamine impurity reference standards, especially the internal standards for NDSRA, from specialized vendors can be time-consuming. Some of the nitrosamine impurity reference standards may be expensive. The future of nitrosamine analysis lies in ensuring patients' safety and drug quality. To achieve this, advancement in analytical techniques like LC-MS-MS and GCMS are crucial for enhancing detection capabilities. Ongoing research is essential to address emerging nitrosamine impurities and develop robust analytical methods. Moreover, advancements in data analysis tools will aid in handling complex datasets generated from nitrosamine analysis. By investing in these areas, we can safeguard the safety and quality of pharmaceutical products. This is the end of my presentation. Thank you for your time and attention.